all the genes and what percent do they contribute to the disease. Okay? And it's tricky because each one makes a very small contribution. It's hard to know what is the, for a particular cancer, which gene is the one that's really driving this cancer right now. It's not like sickle cell anemia or cystic fibrosis where it's one gene where you get the mutant allele, you got it, right? Here you got a whole bunch of alleles you've got to try to track down, different genes track down and see which one is maybe the one that's driving this particular disease state. Again, I'll repeat myself again, many polygenes have an environmental influence. Okay? There's those quantitative diseases like cancer and heart disease. So, sometimes geneticists tend to forget about the environmental influence on a phenotype. They tend to think, well, you got this gene, you're guaranteed to have this phenotype. We know that's not true. Like, for example, here's an Arctic fox whose coat color changes with the seasons for the obvious benefits, because in this case you've got the synthesis of melanin is temperature sensitive, right? The alleles are not expressed when the temperatures are cold, but they are when it's warm for the obvious benefit of the animal. So we have to, when we talk about phenotypes and character states of organisms, we have to remember that there's a genetic component which sort of sets the range of possibilities. Like I realized early as a young teenager, I was never going to be in the NBA because I had this problem with my height. Okay, I just wasn't talented to overcome that, like some people. All right. So we can't forget the environmental component. Here's an example from plants. This is a plant we find in California called Achillea millefolium, called yarrow. You take cuttings from the same plant, go to different altitudes, 3,000 meters above, so the high Sierra, maybe the coastal mountains, and here near sea level, the plant grows to different heights. Okay? So the environment influences, so environments do influence phenotypes. You have to remember that. Okay, and that's our last slide for chapter 12. Any questions? Anybody have a question about chapter 12 before we started at chapter 13? So Mendel's our introduction to genetics. As we go through the next chapters, now, if you think the course has been tough, you ain't seen nothing yet. Because now we're headed toward molecular biology, and we're going to ramp everything way up in complexity here. Okay? Yeah?
For the last part, was the high elevation yarrow smaller?
Let's go back and see. A lot of half-food plants are smaller because they try to maintain appropriate temperature as best they can. I don't remember. Let's see. No, the mid elevation were the smallest. Huh. But at sea level, they were the most robust. Looks like to me, biggest flower heads. All right, so let's head toward more modern biology here. Talk about chapter 13, which really is introduction to genes and chromosomes. Okay. And I won't get through this chapter today. I'd like to, but I don't think I can go that fast. Have you understand everything? I'll give it a shot though. Just so everyone's on the same page here, we are now entering chapter 13. Chromosomes. I think chapter 14 will be introduction to DNA structure. Chapter 15 will be on. Chapter 14 will also cover DNA replication, which is quite complicated. We'll talk about repair mechanisms, recombination, how important they are. Chapter 15, we look at expression of DNA. Then we get into gene regulation, a very complicated topic. Then we're going to get into bacterial viral genetics. So we got some complicated topics coming here. But really, it's foundation stuff. It's stuff you got to have as a foundation to be a modern biologist. Okay, let's back up and revisit our hero so far, Gregor Mendel. This is the front page of the paper he published in 1858. He published, he did 15 years of peat. His paper was based on 15 years of peat breeding studies, 15 years worth of work. He published the paper, gave a few presentations near his hometown of Brno, Czech Republic. I think it was part of the Austro-Hungarian Empire at the time. He made no lasting impact on any of the people that were familiar with his work. I'm sure it was a disillusionment for him. and he quit science and then became the abbot of his monastery. When he died in 1884, I told you most of his notebooks were burned and destroyed. It was just clutter to get rid of. I'm sure he would be surprised today, unless he's looking down somewhere, that he's one of the two 19th century biologists that we still talk about. And again, what's his main contribution? Why is he important in terms of the science of genetics? His main contribution is inheritance is particulate. Okay, that these particles of information, of which he never had any clue about their actual physical nature, are indestructible particles that carry information from parent to offspring. Okay. All right. Well, and of course that these particles that they are paired in individuals because you have two parents and they segregate in sex cells and repair at fertilization. So he predicted things like meiosis, which weren't known until years after his death or described. Okay. It was the early 1880s that mitosis and meiosis were first observed. Right about the time that he died in 1884. So right about that time, I don't know if he was aware of that work, but the first observations of mitosis and meiosis, they observed these things to appear and dance around and separate. And scientists noticed that, gee, when these particles appear in meiosis and segregate, they're behaving like these particles that Mendel predicted. Now, Mendel's work in the 1880s was unknown, but in 1900, three Europeans, I'll write their names down for you, De Vries, Correns, and Schermach. 1900, they did a breeding study. I don't know what plant they used. I'm not sure it was pea plant. I don't remember. They did a breeding study, and they came to the same conclusion as Mendel, that inheritance was particulate, that particles were paired in individuals and segregated at meiosis and repaired at fertilization. And when they went to write the paper, they rediscovered Mendel's work. They said, whoop, we've been scooped by 30 years, and they resurrected Mendel to the scientific world. Instead of trying to hide it and take credit, they gave it to the man who deserved it. So they resurrected Mendel as the founder of this idea of particulate inheritance. Okay? And people begin to notice a correlation between what Mendel predicted should happen, these particles in a diploid would segregate at meiosis and repair at fertilization, with the behavior of chromosomes in meiosis. And that led to an American geneticist named Walter Sutton to propose his theory of chromosomal inheritance. And here's the important part of this theory, that the association of paternal and maternal chromosomes in pairs, their subsequent separation during the reducing division. What's the reducing division called? Remember, there's two types of division in eukaryotes. There's mitosis and meiosis. Which one is the reducing division? Meiosis may constitute the physical basis of the Mendelian law of heredity. So they're saying we've observed that chromosomes behave like Mendel predicted these particles. So his theory, his explanation, is that chromosomes are the particles of inheritance. Okay? So it's known as the theory of chromosomal inheritance, proposed in 1902, 2 years after his name was awoke to the world, scientific world. Okay? And again, it's based on observations that these chromosomes behave like Mendel's particles. They're living sexual organisms. You have a pair of identical chromosomes. They segregate at meiosis and repair, just like Mendel predicted. Okay? Now, that observation needs some more experimental support. And the experimental support that's going to confirm this law of chromosomal theory of inheritance comes from a lab at Columbia University in New York City by one of the great early geneticists of the 20th century. This is Thomas Hunt Morgan, who later was a professor at the University of Kentucky, where I did a year of study there. These are some of his top students in what they call the fly room at Columbia in downtown New York City, because what Morgan chose as this study organism was not a pea plant, but was the fruit fly, Drosophila melanogaster, which is still an important model organism in genetics. In fact, the World Center for Drosophila Genetics is at UC Berkeley. And many of the key discoveries in genetics come from studying fruit flies. By 1920, they developed a gene map of chromosomes in fruit flies before they knew what genes actually were. So these guys really spearheaded, led the path into modern genetics in the early 20th century. And they're still important organisms today in terms of like cancer research. Because it turns out most of the genes in Drosophila are the same ones in you and me. All animals have pretty much the same genes. You've got a few more than Drosophila because they're a little more complex. But most of the basic chemistry of how Drosophila works as an organism applies to us. So you can study cancer genetics in Drosophila and it applies to humans. So they're valuable organisms. What makes them such an important model animal? Because they have a short lifespan of about 2 weeks. They're easy to grow. You smash a banana in a jar, you can grow them easily grow them. And you can produce all types of different mutants, okay, the different characters like eye color or wing shape, because they would take them down to the local dentist and have the dentist x-ray them for them, produce mutants, and then do the crosses and study the crosses, and they were able to build gene maps. They want to confirm that genes actually are on chromosomes through a very important observation that I'll describe now. So they experimentally confirmed with this study that chromosomes do carry these particles of Mendel. And that was because one day in their colony, they noticed that fruit flies just don't have a red eye. They found a wide-eyed male. Okay? In their colony. It was a natural mutant. Okay? So they had a red-eyed, which is the wild type. Ooh, it's wild. Ooh. Okay. And they crossed her, this wild-eyed, love a wild-eyed female. Ooh, yeah, now you're talking. With a wide-eyed mutant male. Okay? Now she's a true breed because